Right, so we've talked a bit about the basics of protein formation. We've gone from a sequence of, a collection of 20 different amino acids. They've formed a polypeptide chain, and they've started to form simple three-dimensional structures, which is the secondary structure. And I want to talk now about how they start to form more complex three-dimensional shapes. And the final three-dimensional shape of a polypeptide chain is the tertiary structure. So it's folding in a way that's given a final three-dimensional shape. In addition, we will talk a bit about the quaternary structure, and it's just simply expanding on the three-dimensional tertiary structure by being comprised of more than one polypeptide chain. So the whole tertiary structure is where it's just one single chain that's folded initially into secondary structures, and then folded again into a more complex three-dimensional structure. And then when you add more than one, you get a quaternary structure, so even more complex proteins. And partly to recap what we've talked about, and to help us understand how this process occurs, we'll just go through the process again. So we start with our chain of amino acids. So there we go. Each of these blocks, it's an amino acid, and they're bonded together, and we've got our polypeptide chain here, and all to the different sides. Then we've got our different R groups, our different groups with a huge amount of different functional groups on, huge amount of different properties, and they interact with each other different based on the chemical properties of the different amino acids, based on the different R groups, and with hydrogen bonding to form secondary structures, very simple three-dimensional shapes. Or draw an alpha helix here. I'll draw a few. In fact, here's another one. So our polypeptide chain here has just coiled itself into a few helices there. Now, how do we jump from this? We've talked about how we get from a single chain to a slightly more complex three-dimensional shape, but how do we get there into what can be quite complex folded molecules? Well, we'll just go back to sort of chemistry basics, really, and the idea that chemistry is just chance. It's probability, and it's all based on the probability of molecules colliding and chemical reactions happening, and it's all based on the path of least resistance, if you like, or if you're being a bit more technically correct, the more thermodynamically stable path. And a big leading or driving factor for the next step is in proteins is the hydrophobic and hydrophilic properties of the molecule. As you'll remember, we talked about when we're talking about the R groups, the idea that some of these R groups are very have got polar molecules on, they interact with water, whereas some of the other types of R groups, they're non-polar, they're more hydrophobic, and proteins make use of these properties when allowing them to fold in these final stage, when are able to form their tertiary structure. It's easy if I think I draw it on here for you. So if you picture, these are our secondary structures here, and they form in a way such that they're amphipathic. We'll talk about a bit more when we talk about cell membranes, but the idea that they've got different hydrophobic and hydrophilic groups on opposite sides. So on this side, for instance, we've got I'll use pluses to represent R groups and so on that are hydrophilic, that interact positively with water. So on this side, they're all hydrophilic. Whereas perhaps on the other side, more of the molecules are hydrophobic, that they don't interact well with water. There we go. So I'll use the negative symbols to show hydrophobic. So not polarity, but hydrophobic properties of the water. Now, if we bear that in mind, we bear this idea of path of least resistance in mind, then in aqueous solution, in water, which is what the body is, what the conditions of the body are, then all these hydrophobic groups are going to want to come together, going to join each other to get away from this water environment. And what that happens is it simply causes folding of these tubes here. So there we go. So here's our alpha helix, another one, and I cause them to fold together in a particular way. So I'll draw the groups on here. Here's our tertiary structure, so 3 for tertiary, and you can see here we've got all the hydrophobic groups are now away from the solution. They're collected together. It's a more thermodynamically stable arrangement, so it's more likely to happen, again just based on this idea of probability. And all the hydrophilic groups are now on the outside in the water, in the water environment of the inside of the cell, and this is the tertiary structure. Now we've gone, if we go back to the start here, we've got a single chain, we've got a chain of amino acids, we've got developing of the smaller secondary structures with simple helix shapes or beta pleated sheet shapes, and finally we've got the folding into more complex shapes. What's interesting here is that if you consider this has happened with the hydrophobic groups on the inside because it's occurring in the water environment. The opposite is also true. For instance, if you think about proteins that are existing inside cell membranes, we'll talk about cell membranes in a later video. That is an environment that is entirely lipids. You've got surrounded by the lipids of the cell membrane, the phospholipid bilayer. And in that case, the opposite is true, because you're in a lipid environment. You want the hydrophobic groups on the outside, whereas you want the hydrophilic groups coming together, for instance, around a pore in the centre. And we'll talk about how that works in a later video on membranes and membrane channels in particular. So hopefully you've got a good idea then of the stages of primary proteins, which is just a polypeptide chain, the smaller secondary structures, as we talked about here, with developing of simple structures, and how that it uses sort of hydrophobic and hydrophilic interactions to form more complex shapes, and the final shape, well, not necessarily the final shape, but often a final shape, which is a tertiary folded protein. And the final one to just think about is the what we'll call it the quaternary structure. So 4 for quaternary, and there's not much else to understand about for quaternary structure really, except that this is all just one chain. This is one long chain. A tertiary structure, you've just got one long chain of amino acids, one long polypeptide that's folding together into a three-dimensional shape. And the quaternary structure is just when you add several chains together. So quaternary, so more than one chain, one polypeptide. And there's a huge variety of possible possible proteins here. For instance, hemoglobin. You've got several different subunits that come together to form the final hemoglobin molecule. And this system here just helps. Hopefully, we've got a good idea of how proteins form, and I'm going to talk about in some later videos about the different uses of proteins and how they're utilised through the body.